the heart rate, increase in the lipolysis. For beta-2, we have beta-2, we have increase in the vasodilation, increase in the glycogenolysis. For alpha-1, we have vasoconstriction. And for alpha-2, it is mainly inhibitory in nature. So paano niya yan, how will it happen? When the body experiences stress. So it is already given, given na the animal is stressed, so it will, during the stimulation of these particular receptors, so this is usually happening in the stimulation or the innervation via the sympathetic nervous system. So when the animal is experiencing stress, so tama yun that the body will be able to increase yung heart rate, increase the respiration, increase sweating, decrease in the digestion, and others. So it will this will happen during periods of exposure to extreme stress or exposure to a frightening experience. But on the molecular level, before this will happen, there should first be binding. So tama itong sinabi ni Mr. Ricale, binding to specific ligand. So ano yung ligand na yon na we are referring to? There should first be binding of a specific ligand. What is that ligand that will be important in order to achieve this? So that refers to our catecholamines. So ito yung ating Ne at saka E or epinephrine. Ne stands for epinephrine and E stands for epinephrine or adrenaline. So for the alpha-1 receptor, so as you can see here, in the alpha-1 receptors, the norepinephrine is considered to be more active. It is more potent in the alpha-1 receptor as compared to the epinephrine. So it has more affinity. It is more potent to the alpha-1 receptor. It is more powerful to the alpha-1 receptor as compared to the epinephrine. So that, for example, when epinephrine will bind to, when epinephrine will bind to the arterioles, so norepinephrine will be able to induce vasoconstriction at a higher magnitude as compared to that of epinephrine. So for example, another example is when norepinephrine will bind to the nicotinic receptors or the alpha-1 adrenergic receptors that is located in the pupil. So norepinephrine will induce more mydriasis, meaning there will be higher magnitude of pupillary dilation as compared to epinephrine. For alpha-2 receptors, in the alpha-2 receptor, epinephrine is considered to be more potent as compared to norepinephrine. So for example, when epinephrine will bind to the presynaptic neuron of the sympathetic nerve, epinephrine will induce a more profound inhibition of the norepinephrine release from the presynaptic neuron as compared to norepinephrine. For the beta-1 receptors, epinephrine is considered to be equal in potency in terms of their activation of the beta-1 receptor. So for example, when epinephrine and norepinephrine will bind to the beta-1 receptors that is located in the heart, so their binding would be equal. The activation of the heart as a result of the binding of epinephrine and norepinephrine will be equal in magnitude. So for example, when epinephrine will bind to the beta-1 receptor, the magnitude of that particular binding, the effect of that particular binding is equal to the magnitude when norepinephrine will also bind to the beta-1 receptor. So pareho lang sila ng effect or potency to that particular receptor. For the beta-2 receptors, no, epinephrine is considered to be 10 times more potent than epinephrine in terms of its activation of the beta-2 receptor. So for example, when epinephrine will bind to the bronchioles, no, the beta-2 receptors in the bronchioles, so yung ating epinephrine, it is 10 times more potent to induce bronchodilation as compared to that of norepinephrine. Okay, so next slide. So we have here the two major groups of our adrenergic receptors. We have our alpha and the beta receptors. So as you can see here, the degree of potency of this particular ligand or catecholamine is indicated by the plus sign. So 3 pluses means that this particular catecholamine is more potent. It will induce the activation of that particular receptor as compared to only 1 plus. 1 plus. So therefore, because norepinephrine has 3 pluses, so we can say that norepinephrine in the alpha receptor is more potent than epinephrine in the activation of the alpha receptor. So this alpha receptor will subserve smooth muscle contraction. So for example, when norepinephrine will bind to the smooth muscle of the blood vessel or the arteriole, the norepinephrine will be more potent in inducing vasoconstriction of the arteriole as compared to epinephrine. For the beta receptor, particularly the beta-2 receptor, will induce relaxation. So relaxation of the smooth muscle. So it is said that epinephrine is more potent than norepinephrine in the activation of the beta receptor or the beta-2 receptor. So that when epinephrine will bind to the beta-2 receptor in the bronchioles, epinephrine will be 10 times, or it will be more potent to induce the relaxation of the bronchioles as compared to norepinephrine. So therefore, we can say that in the alpha receptor, norepinephrine is considered to be the more potent agonist, or it is the more potent catecholamine to that particular receptor. For the beta receptor, in the beta receptor, epinephrine is considered to be the more potent ligand or agonist in the beta receptor. So aside from the potency of the catecholamines to the particular receptor, so we said that norepinephrine is more potent in the alpha receptor, epinephrine is more potent in the beta receptor. So that is one factor that will tell us whether this particular organ, for example, will have a dominant effect that is induced by a beta receptor, or a dominant effect that will be induced by the alpha receptor. So an example of this is the organ, for example, the smooth muscle. So the smooth muscle has both beta-2 receptor, and it has also alpha-1 receptor. So as we all know, the alpha-1 receptor will induce the smooth muscle contraction. The beta-2 receptor will induce the smooth muscle relaxation. So they have opposing effect. So the question now is, sino ba yung masusunod when it comes to the effect that will be induced by this particular catecholamine? So ano ba yung magiging dominant na effect? For example, when epinephrine will bind to this particular receptor, and epinephrine will also bind to this particular receptor. As we all know, epinephrine has affinity. No, it has the tendency to bind to both receptors, alpha 1 at saka alpha 2. But the only difference is that the epinephrine is more, it has more binding affinity, or it is more potent to the beta than the alpha receptor. So in this figure, this is our smooth muscle, our vascular smooth muscle. And we have here our beta receptor. And in this figure here, yung kulay pink, this is our alpha receptor. So as you can notice here, there are fewer beta receptors, beta 2 receptors, as compared to the alpha 1 receptors. So there are about 13 alpha 1 receptors as compared to 5 beta 2 receptors. So when the concentration of the epinephrine is lower in the blood, at low epinephrine concentrations in the blood or in the plasma, the beta 2 receptors will be occupied because these receptors have higher affinity for epinephrine. So going back to our discussion a while ago, epinephrine is considered to be, it has more affinity, or it is more potent to beta than the alpha receptors. So when the concentration of epinephrine is lower in the blood, so mas pipiliin niya ngayon yung receptor where it has more affinity. So yung receptor na yon is ang ating beta 2 receptors. So when epinephrine will bind to the beta 2 receptors, so it will produce the relaxation of the smooth muscle. How about when there is a high epinephrine concentration in the plasma? So both of these smooth muscle contain alpha 1 at saka alpha 2, a beta 2 receptor. But alpha 1 receptors are more numerous as compared to beta 2 receptors. So at high epinephrine concentrations, the alpha 1 receptor would be occupied because there are more of these receptors, and the effect at high epinephrine concentration is vascular smooth muscle concentration. So as we all know, epinephrine has affinity for both receptors, beta 2 and alpha 1 receptors. But epinephrine is considered to be, it has more affinity to beta 2 receptors. But because the concentration of epinephrine is higher, so after binding to the beta 2 receptors, the excess epinephrine which has not bound to the beta 2 receptors will now bind to our alpha 1 receptors. But because the alpha 1 receptors is more numerous as compared to the beta 2 receptors, so the effect of the alpha 1 receptors will predominate, and that effect is the vascular smooth muscle contraction. So at this event, the effect of a particular catecholamine will not only be dictated, or it will not only be determined by the potency of that particular catecholamine to a particular receptor, but also the number of the receptors that have been occupied by that particular catecholamine. So therefore, when, for example, there are more type of this receptor in a particular organ, that more numerous type of receptor, the effect of that particular receptor will predominate at that particular organ. Now, for example, yung ating blood vessels. So blood vessels, ano yung mas numerous sa ating blood vessels, particularly the arterioles? Is it the alpha 1 or beta 2? Based sa inyong previous na mga topics. Ano ba yung mas numerous sa ating blood arterioles?
Yung alpha 1 po.
Okay. So mas numerous yung alpha 1. So therefore, normally, pag sympathetic stimulation, the arterioles would be contracting. So there will be vasoconstriction. For the heart, the more predominant receptor is, ano yung predominant adrenergic receptor sa heart? Beta or alpha?
Beta.
Okay, beta 2, beta 1, no? Beta 1. So sa heart, no, since beta 1 will subserve increase in the heart rate, so therefore, mas mag-dominate ngayon yung effect ng epinephrine doon sa heart, which is increase in the heart rate and increase in the blood pressure. For the lungs, no, the more predominant one is the beta 2, no, so that sympathetic stimulation to the lungs would cause bronchodilation. So this is another diagram showing the effects of the stimulation of the alpha receptors, particularly on the alpha 1 receptor. So stimulation of the alpha 1 receptor will induce the vasoconstriction, contraction of the pyloric sphincter, contraction of the urinary sphincter, and pupillary dilation. So this pupillary dilation or mydriasis is of course true during adrenaline rush or fight or flight. And the same is true for the urinary sphincter contraction. The individual doesn't feel the urge to urinate during fight or flight. The same is true for the digestion process. There is decrease in peristalsis, and there is also vasoconstriction during fight or flight response. Ano gani yung cause why there is vasoconstriction during fight or flight? Assignment nila lang. For the alpha-2 receptors, for the alpha-2 receptors, so when norepinephrine will bind to the alpha-2 receptor, it will decrease the further norepinephrine release. So in this figure, we have here our postganglionic neuron. But this should be, since this is alpha-2, so this should be preganglionic, preganglionic sympathetic neuron. So our preganglionic sympathetic neuron, so our preganglionic pala doesn't release norepinephrine, so only acetylcholine. So assuming that this is the postganglionic sympathetic neuron, it will release norepinephrine. So ito yung mga bilog-bilog, these are the norepinephrine. So when the neuron will be stimulated, so they will release norepinephrine in the synaptic cleft to bind to the adrenergic receptors on the target organ. So when this norepinephrine will bind to the alpha-2 receptors that are located at the terminal portion of this neuron, so it will induce the inhibition of the further secretion of norepinephrine. So this is classified as a negative feedback mechanism in order to prevent the further secretion of our norepinephrine neurotransmitter. So simplifying the receptors, for the alpha-1, this alpha-1 will subserve contraction of the smooth muscle. For alpha-2, they are mainly inhibitory on the presynaptic nerve terminal. For the beta-1, they are mainly found on the heart and the kidneys. For the heart, it will increase the heart rate. For the kidney, it will induce the release of renin. For the beta-2, so the beta-2 receptor is mainly for relaxation. Beta-3, this is found on the adipose tissue. So this is a diagram showing our beta-1 receptors that are mainly located on the heart and the kidney. So the beta-1 receptors are GPCR, and they are located in the cell membrane. So when our agonist will bind to the beta-1 receptor in the heart, it will induce the increase in the SV or the stroke volume increase in the heart rate, and increase in the cardiac output. So when there is more blood that will be pumped by the heart, it will increase the blood pressure. In the kidney, when the norepinephrine, for example, will bind to the beta 1 receptor in the kidney, it will induce the release of our renin. And as we all know, renin is important in the RAS system, or the renin angiotensin aldosterone system. This system is important to correct low blood pressure and to increase yung blood pressure. For the beta 2 receptors, they are found in the lungs, in the liver, in the bladder, and in the blood vessels. So in the lungs, no, when norepinephrine will bind to the beta 2 receptor in the lungs, it will cause bronchodilation. When it will bind to the receptors in the stomach, so it will decrease the peristalsis. For the liver, it will increase the release of glucose from, for example, from glycogen and from other non-carbohydrate sources. For the bladder, it will decrease urination. The responses of the target tissues to catecholamines. So this is a summary of the different responses of the target tissues to. So in the liver, in the adipose tissue, and the skeletal muscle, so it will induce the glycogenolysis, or the breakdown of glycogen, lipolysis, breakdown of fats. Gluconeogenesis is the formation of glucose from non-carbohydrate sources. For the pancreas, no, it will, pag yung ating catecholamine will bind to beta 2, so it will increase the insulin secretion. Pag alpha 2 naman, so since alpha 2 is inhibitory, so it will decrease. For the cardiovascular, of course, it will increase the heart rate, pag beta 1. And pag beta 2 naman, there will be vasodilation. But as we all know, yung predominant na receptor, adrenergic receptor sa heart is beta 1. For the bronchial smooth muscle, the predominant receptor is beta 2. So this is relaxation, GIT. For beta 2, there will be decrease in contractility. For the urinary bladder, alpha 2 is mainly for inhibitory or contraction. For the beta 2, it is for relaxation. There are different factors that will stimulate catecholamine secretion. So one of the main factors that will stimulate the catecholamine secretion from the adrenal medulla is the hypoglycemia, or the low blood sugar. This is the main physiological factor that will influence the release of catecholamines. So in this situation, epinephrine secretion is stimulated by decrease in blood glucose concentration. Another factor is the decrease in the blood pressure. So decrease in blood pressure will also stimulate the adrenal medulla to secrete epinephrine. So catecholamines are important for the maintenance of the blood pressure. Another is decrease in the body temperature. This is also another factor that will induce the release of epinephrine. So one of the main abnormality or disease that is predominant or common that is usually found in the adrenal medulla is Pheochromocytoma. So this is a tumor that is found in the chromaffin cells of the adrenal medulla. So as we all know, the chromaffin cells are found in the medulla, adrenal medulla, and they are responsible for the secretion of our catecholamines. So they are most are malignant. So when we say malignant, it is cancerous in nature. And this this particular tumor no, can produce the epinephrine or epinephrine and dopamine. So there can be metastasis at the time of the diagnosis of this particular disease in dogs. So the metastasis can occur in the different lymph nodes, particularly the sublumbar lymph node. There can be metastasis in the liver, lungs, bone, and the CNS. So 60% are diagnosed at necropsy, and the hallmark clinical manifestation is malignant hypertension. So when we say malignant hypertension, that is a condition wherein there is a very high blood pressure that comes suddenly and quickly. No, so there is malignant hypertension. So this is because this can be attributed to the fact that the adrenal medulla, particularly the chromaffin cells, these are important no, for the production of catecholamines. And catecholamines will increase yung blood pressure. Okay. There is also tachyarrhythmia, or there is, when we say tachyarrhythmia, that is the irregular increase no, in the heart rhythm. So this can cause, for example, the weakness in dogs, exercise intolerance, and collapse. Another clinical manifestation of pheochromocytoma is bleeding diathesis. So when we say diathesis, that is the tendency to bleed or bruise easily. This is because these adrenal tumors, they tend to bleed easily. They tend to bleed. That is why there is this what we call bleeding diathesis. There is also hyphema. When we say hyphema, that is the presence of blood in the aqueous humor of the eyes, aqueous fluid of the eyes. There is also bleeding from the gums and CNS. Okay. There is also PUPD or polyuria, polydipsia in 29% of the cases. And tumors in the caudal vena cava, ascites, the accumulation of fluid and peripheral edema. For the CBC, serology and urinalysis, these are non-specific. And for the abdominal imaging, the abdominal mass may be seen on radiograph or ultrasound. So the abdominal mass are, of course, mainly located on the adrenal portion that is cranial to the kidneys. The adrenals may be enlarged and mixed with patterns of echogenic pattern effacement. So these are technical terms in radiology. For cytology, FNA of the adrenal masses is not recommended. So FNA means the fine needle aspiration. So any adrenal tumor has a propensity to bleed. Catecholamine storm is possible with pheochromocytoma. So when we say catecholamine storm, that is excessive release of catecholamines from the adrenal tumors. So these are the characteristic cellular patterns of when you are going to conduct a cytology of the tumors. Fine chromatin and inconsistent nucleoli. We also have the plasma-free metanephrines. This can be used to measure the plasma-free metanephrine or fractionated urine metanephrine. So this is much higher in people and dogs with pheochromocytoma. So what is this metanephrine? So metanephrine is the breakdown product of our catecholamine, epinephrine and norepinephrine. So this requires special handling of the sample to determine the plasma-free metanephrine. So because this is collected in the plasma, so this is done through serology. So other substances such as, or other drugs such as phenoxybenzaimine. Phenoxybenzaimine is a drug that is an antagonistic drug, in the alpha receptor, corticosteroid, and tricyclic antidepressants can lead to false uh, positive result, to pheochromocytoma. So they can falsely increase the level of metanephrine in the plasma. So for the treatment, adrenalectomy is the treatment of choice. So you're going to remove a portion of that adrenal gland. Perioperative mortality is 50% and pre-treat with phenoxybenzaimine. So this phenoxybenzaimine is important, to reduce the mortality due to hypertension. So treat hypertension. If they survive, the mean survival is 1 year. So any questions with our adrenal medulla?
Doc, i-clarify ko lang po yung yung binigay mong assignment sa amin bago lang.
Okay. Ano yung assignment? So hindi
Kasi
naman yan
yung hindi
talaga.
namin nasagot.
You need na to, ano naman yan.
Ay,
You need
okay.
na to submit. Pero sa inyo na lang,
Sige
you
po.
can
Sige po.
research that on your own. Ano yung cause na. So, let's proceed na. Let's have just an introduction to pancreas. Kasi meron pa naman tayong time. 15 minutes. So for the pancreas, the pancreas is another endocrine gland. The pancreas is actually both an exocrine gland and an endocrine gland. So it is composed of the right and the left lobe. So in gross anatomy, we have learned that the pancreas is composed of two lobes. We have the right and the left, and a body which is located in between. So the right lobe is associated with the proximal duodenum, while the left lobe is associated with the stomach. So this is the diagram of the relationship of the pancreas to the surrounding abdominal organs. So this one here, this is the left lobe of the pancreas, and this is associated with the greater curvature of the stomach. For the right lobe, it is associated with the proximal duodenum. This is also another diagram showing the pancreas and the associated landmarks. So this one here refers to the left lobe of the pancreas, and this one here is the right lobe of the pancreas. So microscopically, the pancreas consists of lobules of glandular tissue surrounded by fine connective tissue. So it has exocrine and endocrine components, and majority of these components are exocrine in function. So 98% of the pancreas is considered to be under its exocrine component. So the lobules are composed of acinar cells, which are responsible for synthesizing the digestive enzymes. So this is the exocrine function of the pancreas to produce digestive enzymes that are important for digestion. Ano gani yung kaibahan ng ating exocrine at saka endocrine? Endocrine pancreas. Ano yung kaibahan nila? Dalawa. yung presence ng ducts. No? So yung exocrine, it has ducts. No? So in the case of pancreas, we have the major and the minor pancreatic ducts. And yung exocrine, hindi siya nagpo-produce ng hormone. So for the endocrine pancreas, so this is only about less than 2% of the pancreas. It is composed of islands of polygonal cells that is known as islets of Langerhans. It forms anastomosing cords and intimately associated with acinar cells. There are 4 distinct cell types in the endocrine pancreas. We have the alpha cells, the beta cells, the delta cells, and the F cells. The alpha cells is responsible for glucagon production. The beta cells is for insulin production. The delta cells is for somatostatin. And the F cells is responsible for the production of pancreatic polypeptide. So this is another diagram of the pancreas showing the exocrine pancreas that is mainly composed of the acini or acinar cells that are mainly exocrine in function. So they produce digestive enzymes that will be transported to the bile ducts. So in this case, we have the common bile duct and the pancreatic ducts. These digestive enzymes will be transported to the duodenum to digest the bolus or the food that is being transported in the GIT. A small portion of the pancreas is composed of the islets of Langerhans or the pancreatic islet, and this is composed of the different cells such as alpha and the beta cells. So this is another schematic diagram of the pancreatic lobule showing the intimate relationship of the acinar cells and the islet cells. So the blue ones are the acinar cells, while this one here are the islet cells. So in between are we have the ducts and the blood vessels. We have here a histologic section of the pancreas showing the islet of Langerhans and the associated exocrine pancreas in H&E stain. We also have here another portion of the endocrine pancreas showing the pale staining groups of islets of Langerhans. So for the exocrine tissue of the pancreas, they are more darker staining in a standard histological section as compared to the islet cells. So this is another depiction of the pancreatic islet showing the different types of cells. Now the beta cells are the ones in the center. They are the ones responsible for the production of insulin, while the alpha cells is the one responsible for the production of glucagon. So the islets of Langerhans was derived from the first person or the first individual which described the pancreas, and he is known as Paul Langerhans. He is a medical student in Berlin, Germany in the year 1869. He was studying the structure of the pancreas. So under the microscope, when he noticed tissues that are scattered throughout the pancreas, he named them as the islets of Langerhans. So the name of the endocrine pancreas is derived from this person. So the major and the most important hormone that is produced by the endocrine pancreas is the insulin. So insulin was first described in the year 1889 by a physician known as Oskar Minkowski. So together with, in collaboration with his companion or his fellow, Joseph von Mering, they removed the pancreas from a healthy dog to test its assumed role in digestion. So on testing the urine of the dog, they found sugar in the dog's urine, establishing for the first time a relationship between the pancreas and diabetes. So as we all know, in diabetes, there is the presence of sugar in the urine. So Oskar Minkowski, together with von Mering, they were the first scientists to establish the relationship between the role of the pancreas and the development of diabetes. So they studied a healthy dog and they removed its pancreas. These are another four important scientists that were that have studied, that have discovered, isolated, and purified insulin, the hormone insulin. So the first one is Frederick Grant Banting and Charles Best. They collaborated and conducted experiments in dogs, and their experiments led to the discovery of insulin. We also have John MacLeod. MacLeod provided the laboratory and scientific guidance through the research in insulin. He had active role in the final steps of isolating and purifying insulin. Finally, James Collip played also a central role in isolating and purifying insulin. So in the year 1921, these four scientists were behind the ending the scourge of diabetes by discovering the hormone insulin. So this is Frederick Banting and Charles Best in Toronto Medical Building in the year 1922. They used dogs as experimental subjects to test the presence of insulin in the pancreas. In the year 1922, the first human insulin, or the first human use of insulin was recorded in the person of 14-year-old Leonard Thompson. He was the first person to receive the medication to combat his type 1 diabetes mellitus. So on January 11, 1922, Leonard Thompson, a 14-year-old diabetic boy, so this is Leonard Thompson in December 15, 1922, at the age of 3, weighing only about 15 pounds. He is a diabetic boy with type 1 diabetes. He became the first human to receive insulin when he was injected by doctors at a hospital with 15 ml of a beef pancreatic extract prepared by Banting and Best. However, on January 23, using an improved extract, Thompson had an immediate response with blood glucose becoming normal, ketones disappearing, and obvious clinical improvement. So after the administration of this particular pancreatic enzyme or pancreatic extract to the patient, there is an improvement in the clinical signs, wherein there is the normal, the blood glucose becomes normal, and also the ketones disappeared, and improvement in the clinical condition of the patient. So this was the first proof that a pancreatic extract could indeed treat and reverse the diabetic state of the person. So in the year 1923, Frederick Banting and MacLeod, no, they received the Nobel Prize Award, no, for their discovery of insulin. Another important scientist or biologist that is attributed, no, to the insulin is another Nobel Prize awardee known as Frederick Sanger. So Sanger or Frederick Sanger determined the primary structure of insulin. It was the first protein to have its sequence be determined. He was awarded the Nobel Prize in Chemistry for his work. So that is Frederick Sanger. So insulin is a polypeptide hormone. So when we say polypeptide, it is a protein hormone. It is produced in the beta cells of the islets of Langerhans in pancreas, and it has profound effect on the intermediary metabolism or the metabolism of fats, proteins, and its most important effect, which is the metabolism of carbohydrates. So for the structure of insulin, the human insulin contains about 51 amino acids that are arranged in two polypeptide chains. So there are two chains, the A and the B, and they are connected by disulfide bridges. So the chain A is composed of 21 amino acids, and the chain B is composed of 30 amino acids. And they are connected by interchain disulfide bridge and intrachain disulfide bridge. So when we say interchain, that is the connection between two chains, the chain A and the B. And when we say intrachain, that is within the chain A. So this is the structure of the human insulin. It is composed of two chains. This is the A chain with 21 amino acids and the B chain with 30 amino acids. So all in all, 51. So there is a link between the chain, the two chains, and that is known as the interchain link. That is the disulfide bridge, so two sulfur molecules. And we also have here two disulfide bridge connecting the chain A and the chain B, and we have another chain here, disulfide bridge, that connects our amino acids, cysteine residues in the chain A. So our amino acid chain has an N-terminal and a C-terminal, which is usually fixed. It is usually fixed in different animal species. This is another illustration of the structure of insulin, showing the two chains and the connection between the different amino acids. So the connection is the disulfide bridge. And they are numbered, the A chain, the amino acids that are found in the A chain are numbered from 1 to 21. B chain is numbered from 1 to 23. So since it's already time, so we will just continue this next meeting, and I will just upload our PDF on our page. So again, see you next meeting. So good morning to everyone.
Thank you, sir.
Thank you, Doc.
Thank you, sir.
Thank
Yes,
you,
sir.
sir.
Thank you, sir.